Thank you very much. It's really nice to be here and in some ways a homecoming, which I will describe to you in a little bit. My talk is entitled Placental Origins of Health and Disease from Environmental Drivers to Solutions. And for that solutions piece, that's one of the areas of emphasis that I'm really thinking about now as I continue my career in environmental science. And one of the ways that I have found success and enjoyment in working in this area is to combine in vitro models, in vivo models, and human population research. And I'm going to tell you a few vignettes about the chemicals that I've studied and the health effects that we've looked at, and intersperse in here some of the solutions that we're working on. As Trevor said, I've focused primarily on toxic metals, but in the past few years have expanded into the forever chemicals, perfluorinated. So I'll talk to you about those. And I always find it really interesting to talk to people about how they ended up in a scientific career. So as you can see from the graphic, I knew exactly that I was going to be an environmental scientist from the very beginning. Not at all. I was born in Massachusetts, and then when I was about 3 years old, my parents moved to Coatesville, Pennsylvania, which is not far from here. I lived there until I was 21. When I was 16, I received a Rotary scholarship to live in Brazil. So I lived in Brazil for a year, stayed with 4 different families. So I do speak Portuguese. And then I went to college in Geneva, New York, William Smith College, which I learned was even colder than places I had ever lived before. And so every opportunity I got, I took a semester abroad. I went to Oaxaca, Mexico, where I studied biology. That's actually how I became a biology major. And this is the woman who taught biology on that semester abroad. And I asked her after that trip if she would be my advisor. And she said yes. And then I went to Brisbane, Australia, where I met my husband. So a lot of things can happen on this windy path. I started graduate school at Tulane and I finished graduate school at Yale. And then I went on to MIT, where I put a picture of Leona on the left. And then does anyone happen to know who the picture is on the right? So these are, you'll notice, all women who have influenced my career. Woman on the right happens to be the Princess of Thailand. And so at one point when I was a postdoc at MIT, I was working on genomic technologies, and Leona came in and said, there's a cohort in Thailand and they're potentially being exposed to arsenic, and we could, understand health effects, and I'd love it if you would focus on the genomics of what's going on. And it happens to be a collaboration with the Princess of Thailand. I said, I think I'll say yes to this one. I'll say yes to this one. Okay, there we go. And I love team science, and team science is the way that we have fun, that we inspire each other, that we continue to have groundbreaking research. So I have a picture here of my lab, of the Institute for Environmental Health Solutions at its launch. We're bigger now. And at our last retreat, which thank heavens we're back in person after the, well, I shouldn't even say it, but thank heavens we're generally back in person. So the study that really influenced me was the one that I just told you about, where we focused on a community in Thailand that was being exposed to arsenic prenatally. At this point in the time of science, there wasn't a lot of research on prenatal exposure and the effects on the transcriptome. There was lots, of course, from hundreds of years of understanding how prenatal exposures could influence human populations. But one of the things that was interesting to me, and this picture shows you some of the genes that we identified in that study that appeared to be turned on or turned off by prenatal arsenic exposure. And so I was really fascinated by wondering what was the switch, what was influencing genes being turned on or turned off. In that study, we looked at blood as our sample that we were collecting. If I could do everything over again, and it just wasn't possible. And in many areas around the world, it's not part of the culture to be able to collect placentas. But over the years I've transitioned to focusing on the placenta. And so one of the reasons that I do, and many of us do focus on the placenta, is that it, of course, is critical for protecting the fetus from various things. It's essential for transporting oxygen and nutrients, removes waste, produces hormones that are critical for human development. But then in environmental science, we also know that in some cases it can serve as a barrier, for example, to cadmium. If you're exposed to cadmium, the placenta has a protein known as metallothionein that can prevent, not completely prevent, but protect against some of cadmium transport. But for other chemicals, they go directly through the placenta, and arsenic is one of those. So what that means is a pregnant woman who's drinking water that might be contaminated with arsenic, that arsenic is transferring through the placenta and exposing the developing fetus. And so the way I think about the placenta is as a sensor and transducer, and sometimes I call it a recorder, like in the old days where you would press a button and there was a tape and it would record, is the way that I sometimes think about the placenta. We also know that the placenta is complicated in some ways. It acts like a tumor. When we have successful placental development, the placenta is, we have cells in the placenta that are invading the myometrium. We have specialized cells within the placenta that are critical for that invasion. And in fact, many of the biological pathways that we might see turned on in a tumor are turned on in a placenta that is behaving properly. And we also know, let me see if I can move this bar, maybe not. Oh, yeah.
Hide caption.
Where was it?
Yeah.
Hide floating
hide.
meeting controls.
Sorry, where is
A
it?
couple down, hide floating meeting control.
Oh, yeah. Thank you. Excellent. We also know from probably hundreds of years of research, if not more, that the placenta and its physiology and function are tied to both pregnancy complications and diseases later in life. So an improperly developing placenta is associated with preterm birth, preeclampsia, intrauterine growth restriction, and again, not just the neonatal outcomes, but later life health outcomes. And so that points us to, again, the placenta as a key target organ for origins of health and disease. And some of what we've looked at over time are genes that are turned on or turned off in the placenta and what controls them, whether that's transcription factors or microRNAs or the methylation of cytosines, which has been a large focus of the lab. So the cohort in Thailand that I told you about that I worked on as a postdoc was here, Ranpabun, Thailand, district of Thailand. And this is a map of areas around the world where arsenic is known to contaminate water. So here's one. I'm going to take you and just think in a minute to Lagunera district of Mexico in the Zimapan Valley, where when I started my faculty position at UNC, I launched a pregnancy cohort. And then you have probably heard also about West Bengal, Bangladesh. This is one of the worst hit areas in the world for high levels of arsenic contamination. One of the worst mass poisonings of populations. And then this map doesn't do justice. One of the facts that I'll share with you in the presentation, which is that we have arsenic contamination in many areas of the country as well. And so over the years, again, focused heavily on understanding toxic metals, their influence on pregnancy outcomes, their influence on fetal development. I'm just sharing with you a fairly recent publication of Catherine Bulka, a former postdoc in the lab who's now running her lab in Florida, where Catherine looked at data from the NHANES study, looked at the data on metals from women of reproductive age, asking in how many of those samples metals were detected, and she showed, ranging up to 99% of the study population, and also found results that are not shocking, probably to many of us, which is that there were racial differences in those levels, and that where you live influenced those levels as well. One of the major areas of focus for the Superfund program that I run is on chemical contamination that can occur in private drinking well water. Many of us are on public water supply. Pennsylvania and North Carolina are two states that rank at the top of our country for having a large number of individuals on private drinking wells. Can you raise your hand if you're on a private drinking well? That's interesting. So no one in here. Sometimes especially when I give talks in communities in North Carolina, almost all the hands in the room go up. And so what we know about private wells is they're not regulated by the EPA, that generally you have to know if you live on a private well that it's not regulated. You have to know where to send your sample to be tested. You have to pay to have your sample tested. So it actually is a very potential source for chemical contamination for a large number of our communities in the country. And then as we think about public and private well water, there are considerations that head us toward environmental injustice. For example, populations on well water are generally rural, and the fact that municipal underbounding has happened, which is a historic practice where municipal boundaries can be gerrymandered to exclude Black, Indigenous, and people of color from public water supply. So some of the research that we've done in North Carolina. So let me just describe that 15 years ago, I started my lab at UNC, and I had just published the work in Thailand, and I was meeting with the state toxicologist in North Carolina, and I was mentioning that I was going to launch a cohort study in Mexico, and he said, "You don't have to go outside the borders of North Carolina to find this issue," but it hadn't been published. And the reason he knew about it was, as the state toxicologist, he was receiving the private well reports that were being analyzed at the state. And he knew exactly which counties were high, but again, it hadn't been quantified. So one of my early graduate students worked on characterizing this. And recently, so this was published in the past year, we have updated that initial publication. So we have collected all of the information from the North Carolina Department of Public Health. We had to pull it down. It's publicly available, but what that means is there's a PDF for every well water test that happens in the state. So there was no database. And Lauren Eves is the person who generated that database. And so what you're seeing here is a map of arsenic contamination in North Carolina and a map of manganese contamination. And so one of the things that's fascinating about North Carolina, you might ask, why is there arsenic in North Carolina? For the same reason that arsenic is in New Hampshire and is potentially in Pennsylvania, although I have to do a little bit more reading about that, is because of our geology. So millions of years ago, there were volcanoes in this area of North Carolina, and the arsenic is now there as a result of that. It's naturally occurring result of geologic formations. So the public health implications are, if you're an individual who lives in the central part of the state and you're on private well water, there's the potential for you to be exposed to high levels of arsenic. The EPA standard for arsenic is 10 parts per billion. That means that in public water supply, our public water supply entities are measuring and monitoring to ensure that we're not drinking water above that. In North Carolina, it reaches 800 parts per billion. This is much, much higher than in, for example, in the study in Mexico that I did. And these are levels that you would see in West Bengal and Bangladesh. And these levels, you can see high levels like this in other states in the US. So what is a solution that we can use to address this? First is helping individuals to understand the potential concern. Second is helping them to measure, reducing that barrier of the cost of testing. Third would be trying to prevent exposure. So we are working with communities in North Carolina, those who reach out to us, for example, in Stokes County several years ago, we had one of actually the largest coal ash spill in the country, and the community in Stokes County where that spill had happened reached out to us. And so we helped them to measure chemicals in their water, provided them with filters that can remove arsenic. And then another solution, or heading towards solution, that we're working on is I work with Tracy Manick, who's in maternal fetal medicine, and Heather Williams, who's a pediatrician, where we are testing for Tracy. We've been doing this for a few years, and we have probably 800 participants who are high risk for preterm birth, from whom we are measuring water. If any water is detected to have any of these toxic metals, we reach out and provide the patients with filters. Now, these are tabletop filters because that's what we can afford. To be able to address this issue, one would likely need a reverse osmotic filter, which is probably $300 or so. So for some of our communities, obviously, we do have a price point issue. Another solution that we've been working on is tools to identify populations at risk. So we applied for some COVID supplement funds for our super fund to develop a tool that we call NC EnviroScan, and you can get to it by enviroscan.org. And this, the goal of this tool was to create a web-based platform. So if you, I'm not going to do it now because that never works, but if you click on the map, you can bring up North Carolina information. We've populated it with the NC well data. You can overlay that with demographic data, and you can overlay that with health outcome data. So for example, if you wanted to know, is there a county where preterm birth risk is high, where arsenic is high, and where the population is poor, you can do that with this tool. And we're currently working to see if there are interested parties in providing some funding for this, because Superfund loves entrepreneurial efforts. But most importantly, we're using it to help guide public health initiatives and also provide a tool and a web source for people to identify solutions for this problem. So I'll share with you some of our research on inorganic arsenic. Two people who really informed my thinking as I was, beginning this work on understanding health effects of prenatal metal exposure were Mike Walkies, who passed away in 2022. Mike was at the NIEHS and NCI, and Alan Smith, who is an epidemiologist at UC Berkeley. Mike used mouse models to study arsenic toxicity. So both of these scientists focused on prenatal arsenic exposure, Mike in mice and Alan in human populations. And both of them really helped me to understand that and provide evidence for the fact that prenatal arsenic exposure is associated with later life health outcomes in mouse models related to carcinogenesis, in human populations in terms of dying of both cancer and non-cancer risks. So the cohort that I launched in Mexico, I've called the BEAR study. And the idea here was we knew that there was a population where arsenic was high in drinking water, but there had never been a biomonitoring study. I worked with Gonzalo Garcia Vargas and Mirek Stiblo. Gonzalo was in Mexico and is a physician there, and Mirek has been a longstanding collaborator of mine. He's in the Department of Nutrition at UNC, and Jess was the graduate student at the time. So we recruited 200 women, measured arsenic in their drinking water and urine, and then collected cord blood. And again, I would ideally have been measuring what was in placenta. It was not culturally or actually feasible in the context of this study to collect placenta. But some of the information that we found, I think, spans target tissues. And so I'll explain that in a little bit. One of the major findings from this study was that as arsenic exposure increased in the population in Mexico, the weight of the babies decreased. And this finding has now been shown in populations around the world. So this is an estimate of birth weight decrease in populations in Bangladesh, Chile, Taiwan. It's also been shown, Mexico is our study, and in Oklahoma. So we know that prenatal arsenic exposure is associated with being born at lower birth weight. And one of the questions that I was interested in asking was, which genes may drive that birth weight effect? And are those genes being controlled by CpG methylation? And glossing over several years of work and hundreds of thousands of dollars and a lot of student effort, one of the genes that really rose to the top that we were focusing on was this imprinted gene KCNQ1, which is here. And these imprinted genes, as I'm sure many of you know, don't follow Mendelian laws. Their expression depends on the allele that you inherit, from which of your parents you inherit. And interestingly, KCNQ1, if you're looking at chromosome 11 in humans, falls in this hotspot for imprinted genes. So there are many imprinted genes. You may have heard of several of these. I know Marisa works heavily on this one. And you may have also heard of, for example, insulin growth factor 2 is very famous from studies that were published on populations that experienced in utero exposure to famine resulting from the Dutch famine in the 1940s, where 2008 PNAS paper linked that periconceptual exposure to famine to insulin, decreased methylation of insulin-like growth factor. So one of the things that has been observed across our study and other studies is almost a susceptibility of these imprinted genes to environmental insults. One of the things that's interesting about imprinted genes is that their expression, again, is tied to the parent from which you inherit the allele, and maternally expressed imprinted genes can be growth restricting, paternally expressed genes can be growth promoting. So this has been described as a tug of war between mothers and fathers over maternal resources used by the fetus. The other thing that's fascinating about imprinted methylation marks is that they're being established prior to the zygote being formed. And so when we saw this imprinted gene rising to the top for our Mexico study, we wondered if the exposure window of concern is actually preconception rather than the in utero period. And so this is something that's hard to test in human populations, easier to test in mouse models. And so working with Mirek in the Department of Nutrition and Fernando Pardo Manuel de Vieno in the Department of Genetics, we've now published several papers where we have supportive evidence that, in fact, preconception exposure to arsenic is tied to effects in the offspring. And here we're looking at diabetes as an outcome. And one of the interesting things to know is that KCNQ1, the gene that we identified in the cohort in Mexico, has been tied to diabetes phenotypes. Let me just do I have 45 minutes or so?
So it's gonna have bit of time.
Yeah, okay. Not 45 minutes from now.
Right.
Yeah. Okay. So what are other ways that arsenic could influence the placenta and the developing fetus? And are there ways that we can test that in the laboratory? And so here's where I'm sharing with you some of the research that we have done using cell culture models. So here, Cassie Meakin, a former PhD student, used JEG3 placental cells as an in vitro model to understand gene expression changes as it related to dose-dependent increases in arsenic. And so in this heat map, you're seeing red would be increased gene expression, blue would be decreased. And so we can see that even at the low level of exposure to arsenic, we have several genes that are being turned on. And one of the interesting things about these genes that Cassie selected was that they are all known targets of the glucocorticoid receptor. And we had reason to test this from some work that had come out of Dartmouth Superfund research program in mouse models that suggested that arsenic was an endocrine disruptor acting on the glucocorticoid receptor. So in this paper, we showed that genes that are targets of the glucocorticoid receptor show differential transcription. And then Cassie also showed that that was tied to CpG methylation in vitro. And here's, if we're looking at the GR pathway, glucocorticoid receptor responds normally to glucocorticoid hormones. But what's happening in the case of arsenic exposure is the cells are seeing arsenic as if it's a glucocorticoid hormone. So arsenic alters the activation of the glucocorticoid receptor. It also alters nuclear translocation and finally alters binding to DNA. So each of those steps were something that Cassie focused on in her paper. So here we're showing both through human population work and in vitro models that arsenic is a toxic agent associated with reduced birth weight, is influencing CpG methylation of imprinted genes that we know play a role in diabetes later in life, and that in the mouse model, we can show that a critical developmental window of exposure is preconception. And so here is some of the solution-oriented research that I'd like to infuse. Leah Clark is a former PhD student. She's now doing a postdoc at the Pasteur Institute and keeps texting me fun things in French. She's having a great time. And Leah focused on asking whether or not there were any nutritional factors that could modify the association between prenatal arsenic exposure and that birth weight effect. And she published that folate and vitamin B12 were potential modifiers of that. So we haven't done any clinical trials, with any one carbon metabolism indicators, but Mary Gamble, who's at Columbia and historically part of Columbia's Superfund program, has used that in, for example, in Bangladesh, where she's done active clinical trials and has shown that if you give populations who are exposed to arsenic folate, that it helps them to metabolize arsenic to a greater extent. So anyway, our results suggest that this could be a way that populations could modify the toxicity of the prenatal exposure to arsenic. So the summary for this first vignette on arsenic is here, that we see an effect on lower birth weight, that it's tied through association to CpG methylation, linked to imprinted genes, that preconception exposure is associated with offspring health, and that one mechanism for its toxicity is through GR hormone signaling. We're working right now on determining whether or not that same effect of seeing imprinted genes is apparent in mouse models. And right now, the answer looks to be like yes, and we are seeing the same gene, KCNQ1, in the livers of mice that were exposed preconceptually to arsenic. So we're excited about those results. I'm going to share with you just a little bit of our work in working with forever chemicals, per and polyfluoroalkyl substances. Living in North Carolina is quite something. There seems to be chemical exposure happening all the time. So maybe that's a good thing if you're an environmental scientist, but not a great thing if you are living there, obviously. So we had researchers who discovered through, they were environmental scientists at NC State who were measuring water samples in the Cape Fear River, measuring them for perfluorinateds, including one of the newest perfluorinateds known as GenX. So the researchers found that there were very high levels of these perfluorinateds, including the new ones, replacements for those that have been banned. And amazingly, our legislature funded several of our universities to address this issue. So we received a few $1 million from the North Carolina legislature to launch the PFAS network, or the PFAS testing network. So this was a collaboration between NC State, UNC Chapel Hill, Wilmington, Charlotte, NCANT, and Duke. And I was part of Team 5 with Jamie DeWitt, and there were several other teams. You may recognize some of these individuals as folks who were part of our P30, like Kathleen Gray, who runs the Community Engagement Corps in the P30. And the original team that identified PFAS in North Carolina is shown here, Detlef Knappe, who's at NC State, an environmental scientist. And Jason Surratt in my department led this effort. So what we know about perfluorinateds is that, as with many chemicals, the outcomes that they cause depend on who's exposed and when, and whether or not they may be pregnant. And so what we know about exposures that happen during pregnancy is they can decrease fertility, increase risk of high blood pressure and preeclampsia, and are associated with lower infant birth weight, just as we see with arsenic. And then with animal models, obviously we can get a little bit more to mechanism, and we know that PFAS are associated with immune system damage, liver damage, birth defects, and other outcomes. So in our lab, we wanted to look at first, to assess what were the levels of the perfluorinateds in the placentas of women from North Carolina, and could we understand the mechanism if we exposed placental cells to those. And common PFAS that we looked at across both of these studies were PFOA, PFAS, and GenX. For the placental study, this work was, the first author on this was Jackie Bangma, who was a postdoc in the lab and is now running her own lab at the EPA. And Jackie had done graduate work working at NC State, capturing alligators in North Carolina and collecting their blood and analyzing them for perfluorinateds. I'm always so jealous because there are pictures of Jackie, wrestling an alligator. I don't think I have any pictures that can beat that. So Jackie had a lot of experience in the chemical assessment of perfluorinateds. And she's a great person to collaborate with. If you're interested in looking at perfluorinateds, feel free to reach out to her. So what we looked at were, 26 different perfluorinateds. We were talking about these today at lunch, and there are thousands and thousands and thousands of these perfluorinated. So we looked at a few, 26, but several of those that have been banned, PFOA, PFAS, several of those that were new, like Gen X or Naphthalene byproduct 2. So many of you may know that sometimes what happens in chemical companies is a chemical will be banned, and then chemical companies can modify that chemical to create a new version. Gen X is an example of that. We worked with Tracy Manick at UNC Hospital, analyzing 122 placentas from high-risk pregnancies. Now, most of these pregnancies were being seen at UNC Chapel Hill, and this will be important later when I show you the results. And here I'm showing you the maximum level of each of those that were detected. So we found 11 legacy perfluorinateds. We didn't find any Gen X or Naphthalene byproduct 2. We didn't find any of these new perfluorinateds in the samples that we looked at. And at the time, there were only 3 other studies that had been published that had measured perfluorinateds in the placenta. There are many studies that had measured perfluorinateds in serum of pregnant women. So if we compare our results, which are shown here on the right, to those from China and Scandinavia, you can see PFAS in blue, PFOA, PFUNA, PFHXS. So in several of these were higher in North Carolina than in Scandinavia, less than in the China cohort. But I mean, generally you can see that we have in some cases similar levels to what has been shown. So that was the first, to our knowledge, the first assessment of perfluorinateds in placental tissue in the US. So we were asking then whether or not there were associations between those perfluorinated levels and various demographic factors like smoking, not smoking, or race. And so what you see here is for smokers, they had higher levels of PFHXS. And for those individuals who self-identified as the other race, they also had higher PFUNA. And so this is the paper that we published a few years ago in ESNT, investigated first the levels so that we could know what we were finding in the placenta. And then we also looked at whether or not there was a relationship to preeclampsia or disorders of hypertension. We didn't find anything that passed a P of 0.05, but we did find for PFUNA borderline significance of the association between PFUNA and hypertensive complications of pregnancy. The reason I bring this up is that in several other studies around the country and around the world, the association between perfluorinateds and, for example, preeclampsia is well established. One of the reasons we may not have seen it is, again, we were sampling from Chapel Hill and not necessarily a highly exposed cohort. I would love, and in fact, there are some studies now that have come out from Duke University and NC State, where they've been sampling from populations that are on the eastern part of the state where they have higher levels, and they are seeing some associations. You had a question?
Yeah, I had a quick question.
Yeah.
I guess I was wondering, did you look also at, so you look primarily at high risk, did you look at other not normal risks?
So it's interesting because even though it's a high risk population, there were many of the children in the cohort who were born at term. So there are within that study, there are both term and preterm infants, but we haven't done one that is an entirely term cohort.
I
yet.
just was wondering in general, if there's more of these PFAS or
Oh.
different, in individuals are exposed more, so you would see it in general.
Yeah, it's possible. It is possible. So for this next part of this, John Szilagyi, who was a postdoc in the lab and I think worked closely with Thea, John was interested in helping to identify the mechanism that might underlie this link between PFAS exposure, and preeclampsia. And so he focused on PFAS, PFOA Gen X. And here he used a placental cell known as SV-NEO. These are cells that where you can assess migration and invasion of the cells. And so he used a scratch assay, which is shown here, where you scratch the placental cells, and then over time you're assessing how quickly they heal, if you will. And he is looking here at the migration of the cells in relation to a control, or PFAS, PFOA Gen X. And you can see that for all of these, migration is reduced in the placental cells that have been exposed to these different PFAS. And then he was also really interested in looking at several cytokines and other genes and proteins that we know play a role in inflammatory response. And so here he looked at protein expression of various proteins, several cytokines, chemokines, and several that are known to have altered expression in preeclampsia, showing that in this case, PFAS, the one in blue, seemed to have the greatest effect in cell culture when he was looking at protein expression. I think I'm running short on time. We've done some other work as part of the ECHO consortium, where we are looking at methylation marks in a cohort known as ELGAN. And this, we've recently been funded for the next 7 years to follow the cohort. Would love to talk with anyone who's interested about ways to collaborate on what we're looking at. These were about 1,000 children who have been followed. Sorry, this should be updated to about 18, 19 years. Very well phenotyped for neurological disorders, and their placental samples were banked early in life. Their original recruiting sites are shown here. So Lake Michigan hub, North Carolina hub, and a New England hub. And they now live all over the country. So I have a really great map that I should have put in here that shows that, you know, if we look at their current geographic location, they're all over the country. I have already mentioned that the placenta is really important as a target organ to study. And some of what we've been doing is asking whether or not there are associations between whether genes are turned on or turned off in the placenta, or have increased methylation or decreased methylation as it relates to several later life outcomes. So we're uniquely positioned in this cohort to be able to look at biologic features within the placenta and some of these later life outcomes. And so here's an example of a paper where we've looked at CpG methylation of various genes in the placenta and asked, do any of those, are they associated with a child's cognition later in life? And you'll see here that we find several CpG sites that are positioned within the gene brain-derived neurotrophic factor, where the odds, we're asking whether or not there's odds of a child scoring more poorly on a cognitive assessment later in life. So the way that I think about these results is I say, okay, what might this gene do in the placenta, and how might that influence fetal growth, and how might that be tied to later life cognition? So BDNF, for example, in trophoblasts, is involved in migration and invasion. So if that gene is altered in the placenta, could influence placental growth and invasion. And then several of the other genes that we've identified here. I'm just going to skip over these just to say, Trevor mentioned that we've looked at sexual dimorphism in the placenta. This is a heat map of CpG sites in our ELGAN cohort, where we're stratifying based on whether the neonate was male or female, and then in a replication cohort. to see that there are many genes that are differentially methylated based on sex. You're probably wouldn't be surprised to know that in this study, when we first looked at them, that many of them are on the X chromosome because that's representing the silencing of one of the X chromosomes. But even if we look across the autosomes, we can see that there's differential methylation by sex. And some of these genes are fascinatingly associated with transport. They represent transporter proteins and also immune response genes. So basically saying there's differential methylation by sex in the placenta for immune response genes and transporters, things that we're actively thinking about when we see sexual dimorphism in outcomes. So I'm just going to jump to the end and say thank you very much for inviting me to come back home